Good morning, good afternoon, good evening, everyone. You are welcome to today's lecture. In today's video, we are going to be discussing about chemistry of amino acids and proteins, lecture 1. So, introduction. Let's first understand what amino acids are before we move into the protein profile. So, what are these amino acids? They are the monomeric units of protein. So when we said monomeric units, it means for protein to be formed, you need the building blocks to come together to form the protein. So these building blocks of proteins are the amino acids. They are the basic structural units of proteins. And proteins in all species from bacteria to humans, they are constructed from the same set of 20 amino acids. So there are 20 common amino acids that are the constituents of all proteins that you know, right from the protein from bacteria and also the proteins that are found in humans. So when you look at this structure here, you will see that there is a carbon that is bonded to R group, bonded to carboxylic acid group, which is COOH, and bonded to hydrogen atom, and also bonded to amino group, NH2, right? So this is the typical representation or general representation of an amino acid. So all amino acids have this carbon, we call it the alpha carbon. This is the alpha carbon, which is bonded to four different groups, except in glycine, which the side chain is hydrogen atom. So all amino acids have this amino group, hydrogen atom, carboxylic group. The only difference between one amino acid to another is this side chain, the R group. The R group is the only thing that is differentiating them structurally. So apart from the formation of proteins, amino acids are involved in neurotransmitter transport, and also biosynthesis. Okay, so there are other amino acids that are not part of these 20 common amino acids. Okay, like GABA, which we call gamma aminobutyric acid, is a neurotransmitter. It's not a common amino acid, but they are found in living organisms. Okay, so amino acid consists of the amino group, carboxylic acid, and a hydrogen atom, and also a distinctive R group, which is specific to each amino acid. This R group is the side chain and is specific to each amino acid. The three groups, they are bonded to carbon called the alpha carbon, as I have explained. So the amino acid take part in many types of reaction, but the most important of this reaction is what is the formation of peptide bond. And the formation of peptide bond formation involves the joining of the alpha carboxylic group, the alpha carboxylic acid group with one amino group that is called the alpha amino group of another amino acid with the elimination or loss of water molecule. So amino acids are grouped according to the nature of their side chain. So you can group amino acid based on the side chain they contain, because all amino acid basically consists of these three groups, but this R group is different in each of them. So amino acids, they are weak acids. Their strength is expressed as pKa, which is the negative log of ionization constant. And the net charge on any amino acid depends on the pKa of its functional groups and the pH of the surrounding medium. So the net charge at the end, the charge that you will see in each amino acid depends on what is the pKa of the functional groups that are in the amino acid, and also the environment in which that amino acid, be it acidic environment, neutral pH environment, or alkaline environment. So the isoelectric pH, which is termed as Pi, is the pH midway between the pKa values on either side of the isotonic species. So is the pH value that is midway between the pK values on either side of the isotonic species. So when we look at the chemical nature of these amino acids, we have already highlighted that each amino acid has this alpha carbon, and the alpha carbon is bonded to the R group, which is the side chain distinct for each amino acid. It has a hydrogen atom, the amino group, and also the carboxyl or carboxylic acid group. So when we look at the how these amino acids generally the form in which they exist at low pH, what is the structure look like? How does the amino acid look like? What about at neutral pH or at high pH? So at low pH, the alpha carboxyl group is what? Is fully is COOH is protonated, okay? While the amino group is also fully protonated. It has NH3 at low pH, because that low pH, it means acidic pH, right? So if an amino acid is in acidic medium, it means there are a lot of hydrogen ions. So you should expect the amino group to be what? NH3. So what about at neutral pH? At neutral pH, we can see that the carboxyl group lost its hydrogen atom. So it is in form of carboxylate ion. So, and this carboxylate ion, and we have the fully protonated amino group, which makes this structure to become zeta ion, because there is a pole here, positive, and there's also negative here. So it means at neutral pH, if you look at it, it has NH3 plus and COO negative, which is called zeta ion. So what about at high pH? When you are moving upward towards alkalinity, it means the alpha amino group is now devoid of this one hydrogen. It means it is NH2 rather than NH3 plus, while the carboxylate ion is still remain the same. So at high pH, you can have it to become a generally it is negative, okay? While at neutral pH, it is physiologically neutral, right? It is electrically neutral. While at high pH, it is what? It is negative. While at low pH, it is what? Positive. So why do you think this happens? Is because when at low pH, the medium is acidic medium, and the medium is having hydrogen ions, right? That is why this is protonated, right? So when you are moving towards alkalinity, it means you are adding, you are from base, you are coming from acidic medium going toward basicity or alkalinity. It means you are adding what? Alkali or a base. You are adding a base to it. So when you add a base, then naturally you are what? Adding OH. When you're adding OH, OH will go and engage with hydrogen ions. You remember? So this is why this carboxyl functional group lose its proton, right? So when you continue to add OH, moving towards alkalinity, it means this hydrogen ion will be engaged. That is why it loses its hydrogen ion to become NH2 rather than NH3 in plus in the zeta ion neutral pH. Okay. So the arrangement of the 4 different groups about the alpha carbon confers optical activity on amino acid. If you remember, when we discussed about sugar, we talk about optical activity or optical isomers, right? We talk about L and D isomers, whereby I presented a structure for you. If you can remember, I have explained that also in amino acid, this D and L isomerism also occurs. So here is this, and we are going to discuss about it. So we have the 2 mirror image isomers that are called L isomer and D isomer. But in protein, L amino acids are exclusively found in the protein. That is to say, all the 20 common amino acids, they are in the form of what? L isomers. So they are all L amino acids, okay? But where can we find the amino acid? They also occur naturally. It include free D-serine and also D-aspartate, this D-aspartate, which is found in brain tissue. These 2 are found in brain tissue, and D-alanine and D-glutamate, which are found in the cell walls of gram-positive bacteria, and also D-amino acids in some non-mammalian peptides and certain antibiotics. So, but in proteins, we have exclusively L amino acids. But in bacteria and in some gram-positive cell wall, cellular gram-positive bacteria, and in brain tissue, we can have some D amino acid, okay? So you look at the structure here, they are mirror image. You remember, they are mirror image. We call them enantiomers. They are mirror image of each other. So this is L amino acid, whereby the amino group is at the left-hand side, okay? You have the alpha carbon, the carboxylic acid functional group, and the R group, which is the side chain, and the hydrogen atom. But the amino group is at the left-hand side, so it is L amino acid. When the amino group is at the right side, we have the amino acid. Don't forget that. So many proteins also contain amino acid, which are usually formed by enzymatic modification of an amino acid after it has been incorporated into a protein. Okay, so for example, we can have what we call hydroxyproline. You know, hydroxyproline is an amino acid, okay, that is found in collagen. Okay, so the OH group serves to stabilize the collagen fiber. We have prothrombin, which is a floating protein. It contains gamma carboxyglutamate. You remember gamma aminobutyric acid that I told you about, which is a neurotransmitter. It is also another example of amino acid that are not found in protein, but they do exist in humans. So when we have defective carboxylation of glutamate in this protein, it may lead to what we call the hemorrhage. So we have prothrombin, which is a clotting protein. It's a clotting factor. It contains what? Gamma carboxyglutamate. So when there is a defective carboxylation of glutamate in the protein, we have what it may lead to what we call the hemorrhage. So the action of some hormones is also mediated by phosphorylation and dephosphorylation of specific serine residues in a variety of proteins. So we can say that this amino acid, the residues that are found in these proteins, we can say there is a kind of what we call regulation of their activity by phosphorylation and dephosphorylation. When we serine residues in some varieties of protein is phosphorylated or dephosphorylated, and we can say that the action of this is also is going to be regulated. So let's look at the 20 common amino acid. This is a picture contains all the 20 common amino acid. We are going to come and be classifying them based on the side chain. What is the component? What is in the R group? Okay, so we can have this glycine, serine, threonine, alanine, cysteine, valine, leucine, isoleucine, methionine, proline, phenylalanine, tyrosine, tryptophan, aspartic acid, glutamic acid, asparagine, glutamine, histidine, lysine, and arginine. These are 20. 1, 2, 3, 4, 5. 1, 2, 3, 4. 4 times 5, which is 20. So there are now 20. These are the 20 common amino acids that are found in protein. So we are going to come and classify them based on what is the R group, okay? What do they contain in the R group? So we'll make that classification. And there is a way this amino acid can be represented. We have the full name, which is written here, as in the case of glycine here, alanine here, right? We have the full name, and there is 3-letter notation. For each of these amino acids, they have 3-letter notation. Like glycine is GLY, gly is the 3-letter notation, while for alanine is ALA, for the 3-letter notation. And also they have a 1-letter notation. For glycine is G, for alanine is A, okay? So for phenylalanine is W, okay? So tryptophan is W, phenylalanine is F, while tyrosine is Y, okay? And tryptophan is W, which is the 1-letter notation for each of these amino acids. So we are going to look at them one after the other. So look at the amino acids here, and this is the abbreviation. We can have the 3-letter notation and the 1-letter notation. For glycine, the 3-letter is gly, and the 1-letter is G. Look at it. For phenylalanine, is PHE, the 3-letter notation, and the 1-letter notation is F. Tryptophan, TRP, and 1-letter notation is W. Tyrosine is TYR, the 1-letter notation is Y. So it's very important to sit down and master this. The 3-letter notation, you master only 20 common amino acids, and also master the 3-letter notation, and also master the 1-letter notation. Is very important. Sometimes it's cumbersome to be writing, let's say in protein, it's going to be cumbersome to be writing for a protein that is having all these 20 amino acids. It's cumbersome to be writing all these names. Rather, we can use the 1-letter notation, which is more easily written, okay? Or the 3-letter notation, okay? So it's important to learn them, all of them. So we are going to, there are some series of questions here that we don't have the answers now, but later at the end of this lecture, you should be able to have the answers. So what are the amino acids that have hydrocarbon side chain? What amino acids have hydrocarbon side chain? What amino acids have the side chain that contains hydroxyl groups? What amino acid contains aromatic side chain? So what are aromatic amino acids? What are amino acids that contains basic side chain? So what are basic amino acids? What are amino acids with carboxamide groups, which are acidic amino acids? Okay. What are which amino acid contain sulfur? Okay. So what are the sulfur containing amino acids? We don't have the answer now, but at the end of this lecture, you should be able to come back and answer these questions. So let's look at now, this is now all the 20 common amino acids. If you look at it, it's been divided into 3 groups. We have the electrically charged, we have the polar, and we have the non-polar. So all these amino acids here, they are all non-polar. Glycine, alanine, valine, leucine, isoleucine, methionine, tryptophan, phenylalanine, and proline. They are all non-polar. Why are they non-polar? It's because of the, look at the side chain. Glycine, the side chain is H, non-polar. Alanine, the side chain is CH3, non-polar. Valine, the side head is CH, CH3, CH3, non-polar. Leucine, look at it, non-polar. Isoleucine, non-polar. Methionine, non-polar. Tryptophan is non-polar. Phenylalanine is non-polar. They have aromatic ring, right? It's non-polar. They are non-polar. Okay, proline is non-polar. Okay, so what about polar amino acids? Polar amino acid, we have serine, threonine, cysteine, tyrosine, asparagine, and glutamine. Why are they polar? Look at their side chain, the R group. Serine has OH, polar. Threonine has another CH, okay? CHOH, CH3, look at it, polar. Cysteine has sulfhydryl group SH, which is polar. Tyrosine has aromatic side chain with OH, okay? It has phenol side chain, right? Right? CH2, aromatic ring OH, it is polar. Asparagine, it has CO, right? Amide functional group, CONH2, polar, CONH2, polar, right? So what about electrically charged? We can say the side chain is electrically charged. We have acidic amino acid, which is aspartic acid, glutamic acid. Look at it. They have additional COO, negative COO, right? Carboxylic functional group, which we can say they are what? Electrically charged. Lysine, they have additional uh, amino group fully protonated plus, right? It's charged. Arginine, charged. NH2 plus, charged. Histidine, NH plus, the ring, right? Is charged. So these three, lysine, arginine, and histidine, they are what? Basic amino acid. Okay? So we can have another classification. This is based on is also the other classification also is also going to be based on the side chain, but you can see that if I ask now a question, what are the non-polar amino acids? You can name all these that are up here. What are the non-polar amino acids? You can name all these that are up here. What are the electrically charged amino acid? They are divided into two. We have the acidic, you can name these two, and the basic, you can name these three. Okay, so now on that basis also, you can, the basis on the polarity, on the basis of the side chain, on the basis of the side chain, you can also classify the amino acid. You can say, okay, what are the amino acid, or which and which amino acid have aromatic side chain? You see, you can answer that questions, those questions that we asked. Which and which amino acid have aromatic side chain? You can see tryptophan, phenylalanine, and tyrosine. Now we're not talking about which one is polar or non-polar. We're talking about aromatic side chain. This has aromatic side chain, tryptophan, phenylalanine, and tyrosine. They all have aromatic side chain. When we say what and what amino acid contain sulfur, what are the sulfur containing amino acid? You can see one is here under non-polar and another one here is under polar. So you can see methionine has sulfur and cysteine has also sulfur. So you have methionine and cysteine, which are the two sulfur containing amino acid. We can also see which and which amino acid have OH group in their side chain, or hydroxyl group in their side chain. We can see serine has OH. We can see threonine has OH, right? We can see tyrosine also has OH. You can see, right? So, and we say which and which amino acid has hydrocarbon side chain. You can see them, hydrocarbon. You can see alanine here. You can see valine here. You can see leucine. You can see isoleucine. Okay. Okay, now look at the structures. Basically, look at the structures carefully, and you should be able to answer all those questions, although I have already answered some for you. So what is this R group, and what is the chemical properties of amino acids? So the R group is the one that determines the chemical properties of amino acid. So the charge functional group of amino acid ensure that they are readily soluble in polar solvent, such as water, ethanol, but insoluble in non-polar solvents, such as benzene or ether. Similarly, the high amount of energy required to disrupt the ionic forces that stabilize the crystal lattice account for the high melting point of amino acids. So they have high melting points because of the high energy that is required to disrupt or destabilize the crystal lattice. So amino acids do not absorb visible light. They don't absorb visible light and are therefore colorless. So because of that, amino acid solutions are colorless, except if they are uh, you put another reagent to them, but their solution is colorless. However, the aromatic amino acid, the phenylalanine, tyrosine, and tryptophan, they absorb high wavelength UV light. It means they absorb what? UV light. Why? Because they have the aromatic ring structure in them. And that aromatic ring structure has alternative pi, right? It has alternative pi bond, right? So that alternative pi bond rather brings about the absorption of light that we notice in this amino acid. So these three majorly contribute to the ability of proteins to absorb light in the region of 280 nanometer. So because of this tryptophan, phenylalanine, and tyrosine, because they have aromatic ring structure, they contribute to the ability of proteins to absorb light at what this region of 280 nanometer. The respective R groups of amino acids determine their properties. For example, the hydrophobic R groups that are found in alanine, valine, leucine, and isoleucine, as well as the aromatic R groups that are found in phenylalanine, tyrosine, and tryptophan, typically occur in the interior of cytosolic proteins. So they are hydrophobic because they don't engage with water, so they are embedded into the interior cytosolic proteins. They don't associate with the water molecules, so they hide themselves because they are hydrophobic. So the charged R groups of basic and acidic amino acids stabilize specific protein conformation via ionic interaction or Sol bond. So each functional group of an amino acid exhibit all its characteristics chemical structure. For example, the carboxylic acid groups, they undergo a reaction that includes formation of esters, amides, and acid anhydride, while those for amino acid group including acetylation and amidation and esterification. So let's look at the pi of an amino acid. The net charge on an amino acid, that is the sum of all the positively and negatively charged groups that are present, depend on the pK values of its functional group and the pH of the surrounding medium. So when there is an alteration in the charge on amino acid and their derivatives by varying the pH, it facilitates the physical separation of amino acid and peptides and also protein. So the isoelectric pH, which is the PI, is the pH that is midway between the pK values on either side of the isoelectric species. For an amino acid such as alanine that has two ionizable groups, it has two ionizable groups. So it has pK1 and pK2. So the isoelectric pH is now termed as Pi, which is equal to the sum of the two pKa's divided by 2. You sum the two pKa's and divide by 2, which is 2.35 plus 9.69 divided by 2, which is 6.02. That is the isoelectric pH of alanine. This actually pK values are given in a tabular form in later slides that I will present. So you should be able to understand and cram them, okay, and put them in your head, okay? For functional amino acid, for polyfunctional amino acid, that is amino acid that have many more functional groups, the PI is the pH midway between the pK values on either side of the isoelectric species, isoionic species, okay? So for example, aspartic acid, the PI is 2.09 plus 3.96 divided by 2, which is 3.20. So let's look at the titration curve of alanine, for instance. This is the pH on the x-axis and the equivalent OH at the at pH at the y-axis, equivalent OH at the x-axis. So when the titration starts, if you look at alanine, it is what? At acidic pH, it is the COOH is intact, NH3 is fully protonated because the medium is acidic, right? So when you start titrating, you are adding OH because it's an acid, you are adding OH to it at about 2 point something. That's where you have your what? PK1, right? So at about 2 point pH 2 point something, you have our PK1, and that PK1, before you are adding OH, you are adding OH to that solution. OH to that solution, it means the hydrogen ion that is found in the alpha carboxylic group is engaged in the reaction forming water molecule. Okay, so now when you continue titration, you at pK2, that's when you have the amino hydrogen ion is also engaged as you are adding OH, eliminating water molecule, right? So you have the Pi, which is in between the PK1 and the PK2. This is what isoelectric point, or isoelectric pH. So when we look at the dissociable groups and also the net charge at varying pH, you have at low pH, or at pH that is below the PK1, right? At pH that is below the PK1, we can say we have COOH and NH3 plus, just like the way it is here. At pH below pK, the net charge is about what? +1, right? The net charge is +1. When you have this at pH between PK1 and PK2, right? You have the net charge to be about 0 because you have COO negative and NH3 plus. So this is electrically neutral, right? So at pH above PK2, at pH above PK2, we can say the net charge is about negative, right? It's about negative. Look at it. The PK at pH above the PK2, you can say it is what? Electrically minus, negative, because you have COO negative and NH2. So it is -1. So this is the summary of how this titration occurs. So when we look at aspartate, which is aspartic acid, when we look at it, it follows the pattern. Look at it. You can say the pKa or the Pi is the pKa in between these two Pi, pK1 and pK2. So the Pi is in between, the pH value in between these two pK, pK1 and pK2. Okay, so now when we look at the summary of the dissociable groups and the net charge at varying pH, we can say we have COOH, and NH3 at pH below pK1. At pH below pK1 at this region, this is what we have: COOH for the R group and NH3 positive, right? So we have the net charge is about +1. Then you have COO negative, COOH. Look at it, COO negative, COOH, NH3 plus. At pH between PK1 and PK2, the net charge is about 0. You have COO negative, and NH3 positive. At a pH above PK2, the net charge is about minus, right? -1. And you have lastly, at pH about pKa, the net charge is -2 here, because you have COO negative here, COO negative, and what? NH, COO negative, and NH2. So similarly, lysine also follow the same pattern, as you can see, but the Pi is also between PK2 and PK3. The Pi is between PK2 and PK3. pH value between PK2 and PK3, right? So look at the dissociable groups, different dissociable groups, and at different pH, they have different net charge. Look at it, up to -1. Okay? So now let's look at the pK values. This is a table containing pKa values for the ionizable groups of the amino acid. Look at all the amino acids. They have the pK1, which is the ionizable group COOH. That is the alpha carboxylic group. And these are the various pKs. pK1s for each amino acid. And this is pK2, which is the ionizable NH alpha amino group, NH3 plus. Look at it. Look at the values here. And the pK3, which is the pK for the R group. pK for the R group. Okay? Those that have ionizable group in their side chain will have pK values, but those that does not have, they don't have any pK values because their side group is not ionizable. For instance, glycine, the side chain of glycine is hydrogen ion, so it doesn't have any pK because it's not ionizable. So also alanine is CH3, okay? It's not ionizable, okay? So, but for cysteine, because it has ionizable side chain, histidine and the rest, they have ionizable side chain, they have pKr, which is their pK3. So how does this amino acid come together to form a protein? Because we say amino acids are the what? Monomeric units of protein. So how do they come together to form that protein? They form a peptide bond. They come together and join to form the protein via what we call peptide linkage or peptide bond. So how does it form? How is the formation of this peptide bond occur? The most important reaction of amino acid is what? The formation of peptide bond. We have explained that. This involves joining of the alpha carboxyl group of one amino acid to the alpha amino group of another amino acid with the resultant loss of water molecule. So where you have the alpha carboxylic group of one amino acid and the alpha amino group of another amino acid, they join together to form the peptide linkage, and there is going to be loss of water molecule. So biosynthesis of peptide bonds require an input of free energy, whereas their hydrolysis is thermodynamically favorable. So when you are forming peptide bond, you need input of free energy, but the hydrolysis is thermodynamically favorable. So many amino acids, usually more than 100 amino acid residues, are joined by peptide bonds to form what we call polypeptide chain, right? Polypeptide chain. So an amino acid unit in a polypeptide chain is called a residue. So an amino acid unit that is found in the polypeptide chain, you know, you have one amino acid joined to another amino acid, joined to another amino acid, joined to another amino acid. So in that case, since it is now in polypeptide chain, when you talk about individual amino acid in that, you don't call them amino acid, you call them what? Residue. They are called what? Residue. So a unit of that amino acid is called a residue in the polypeptide chain. Now, a polypeptide chain has different ends. It has the alpha amino group and the alpha carboxyl groups. So there is an end that is having alpha amino group, and there is end that is having a carboxyl end group. So the amino end is taken to be the beginning of the chain. The amino end is taken to be the beginning of the chain. When you talk about, let's say you talk about left and right, the amino group is at the left, which is the beginning of the chain, and the carboxyl group is at the right, at the end of the polypeptide, which is what? To the right, right? So, for instance, in one tripeptide, you have alanine, glycine, and tryptophan in that tripeptide. The alanine is the amino terminal residue, while tryptophan is what? Carboxyl terminal residue. So a polypeptide chain consists of a regularly repeating part that is called the main chain, also called the backbone, and a variable part comprising of the what? Distinctive side chain. So in some proteins, a few side chains are cross-linked by disulfide bonds. Remember, methionine and cysteine have what? Sulfate group. So when there is going to be this linking, cross-linking, because in some protein you have side chains that are what? Cross-linking between one polypeptide to another, there is going to be cross-linking, and what is joining that is what? Disulfide bond, that because it's due to the what? Presence of sulfur-containing amino acid, usually cysteine. So these links are formed by the oxidation of what? Cysteine residues, and the resulting disulfate is called what? Disulfide bond linkage is called cysteine. Okay, so amino acid present in peptides are called amino acyl residues and are named by replacing the ATE or the INE, let's say glutamine, if it is ending with INE, or glutamate, if it is in ionizable form, right? So that suffix is being replaced by a of that free amino acid with what? Al, e.g. aspartyl, alanine, okay? So for instance, when you have a tripeptide like that is having lysine, leucine, tyrosine, and glycine, and glutamine rather. So how do you call it? How do you call this tetrapeptide? You call it lysyl, leucyl, tyrosyl, glutamine. And why did you call glutamine its full name? Is because it's the terminal amino group. Okay, so the alpha amino group or the alpha carboxylic group of that amino acid is not involved in what? Peptide formation. Okay, it's not involved in peptide formation. That is the last amino group in the chain. So that is why it is called its full name. Okay, so in single polypeptide chain, for example, like in myoglobin, we can have a single polypeptide chain. That is to say, we can have a polypeptide chain that is single, or we can have a polypeptide chain that is maybe having two or more peptide chain. Okay, so a single polypeptide chain is found in, for example, in myoglobin. We can have two or more chains which may be what? Identical or different. For example, in hemoglobin, it's made up of two chains of one kind and two chains of another kind. So it has, all in all, it is having four polypeptide chain. Two polypeptide chain are called alpha polypeptide, while two are called what? Beta polypeptide. So this brings us to the end of today's lecture, and I hope you really enjoy it, and you will please try to go through the lecture courseware and also try to ask questions, and also watch the video and ask questions in the forum. So thank you very much. See you next.